Yeah, so good evening. We will start with the d square first. So, as today in mail, I have shared you some files. Yeah, so just to see once. Am I audible?
Sir, the D square file is available in what? Sorry, Word file, sir, not in Notepad form.
Am I audible?
Yes, sir.
Yes, sir.
Yeah.
Yes, sir.
Yes,
So,
sir.
see, first we will start with d square. There are three files we will use. First one is this average. Second one is this cotton. And command line file, it is there in Word. So this is the command for d square analysis along with dendrogram. So I think you don't have this biotools package in your system. So just kindly install. Just check this all these packages whether it is there in your system or not by running library for each and every one. I think this ggplot2 is already there with you. So run this all the four package library. If it is not there, just install it. After that, let me know. After completion of installation and library, let me know. Done. Okay, done. So now, if your installation and library is done, just import that both the files. That is average and cotton. Import both the files. So the both files belongs to same data. Just here in case of cotton, it is replicated data and average is average of that replicated data. So both files are same. So next, up to this it's done. So after that, we need to separate the variables. So now if you will copy paste it, either here name is data. So either if you don't interested in changing the name, just you need to annotate the file. Otherwise, just change here the name. And here 3 is to 13. So here see from third column 3, 4, 5, 6. So in our case, it is 3 is to 6. Enter. So after that, model for MANOVA. So what is mean by what is this MANOVA? What we are doing? Yes. Which type of analysis we are doing?
Diversity.
That
Multivariate
T
analysis.
square is which type of analysis?
Multivariate.
Yeah.
Multivariate analysis.
Correct. Come again.
So multivariate.
Yeah. Yeah. So then what is meant by MANOVA?
So multivariate analysis of variance.
Yeah. So MANOVA is multivariate analysis of variance. So see, for that, I'll copy this first three. I will tell you. So see here the model DV, as we have done the first step here DV. Then next, this MANOVA. This is our first column genotype, first factor. Second factor is our replication. Third factor, our data is equal to cotton. So see, this is our multivariate analysis of variance. Now here we have calculated something like SSD. So if you don't know what is SSD, just simply question
Hello,
mark.
sir. The screen is not visible.
Screen is not visible.
No, sir.
It is visible, na?
Visible, sir.
I am sharing entire screen.
Visible, sir.
Yeah. Yeah. So if you want to know the meaning of anything, just like I want to know SSD. So what is SSD? Question mark and that word. So here see, right side you will get all the details. With that question mark. So that, yes. So then from that SSD, we will calculate the covariance. After that, from that covariance, we will calculate the percent contribution of characters. So see, out of this 4 characters, seed cotton yield is contributing 82% towards this 82.1% contribution of yield towards.
Divergence.
Yeah. So this seed cotton yield is contributing 82% towards divergence or diversity. So like this diagram you will get. You can make table of it or diagram or both. Then after this, we will shift towards Mahalanobis distance. So to calculate this, we need that average file. Yeah. So can anyone can you tell me the meaning of this command? What we are doing? Why this -1 is there?
Correlation.
This is our average file. Why this -1 is there?
So the first column is just showing number one and number two is
Yeah. So here just we need the average of observations. And we have imported the data with first column genotype column and from second column onwards data. So for that, then if you will type D, you will get this covariance. Then now there are two methods. One is torture and another one is modified torture through use of this algorithm. So which method you need to follow, that is based on you. So with this torture method, see how many genotypes are there? 12 genotypes. So these 12 genotypes are classified into 6 different clusters. So here you can see it. Cluster 1, 4, cluster 2, 2, then 2, 2, 1, 1. And this is the cluster distance. So this is torture method. Secondly, modified torture method. We'll see whether any change is there or not. Yeah. So in torture method, we got six clusters. And in modified torture method, we got 5 clusters. The difference is in both the models, just this algorithm. Added algorithm is there. So up to this cluster pattern, cluster distance. Then we'll proceed towards dendrogram. So now here, if you can see the command 1, 2, 3, 4, line number 5th and line number 6th. So just go to this line number 5th and 6th. Here this K is there. This K means clusters. So whatever the number of clusters we got here, so that number we need to put there. So now suppose through torture method we got 6 clusters. So here I will write 6 and here 6. Then average file, and we want the distribution of genotype. So our first column is not genotypes, it is genotype. Okay, so then I will copy this. So this is the dendrogram for torture method. Now just you can match that number of clusters, in which cluster which genotype is there, and in this dendrogram whether it is matching or not, just we will check it. So just see, in cluster 1, 4 genotypes are there, 5, 6, 7, 9. So here you can see 5, 6, 7, 9. These 4 genotypes are there in one cluster. So second cluster is 4 and 11. So here you can see genotype 4 and 11 is there in one cluster. Next, 3 and 8. So here you can see at last 3 and 8. Next is 1 and 10. So here 1 and 10. Next is 2. Yeah, here it is 2 alone. Then last is 12. Where is 12? Here it is 12. So likewise, clustering pattern, cluster distance, and dendrogram. So here
Hello,
that what we will call that octagon, that diagram will not come. Like it will be the, I think Windows Stat is giving that diagram, na? Windows Stat is, I think, giving that octagon. But if you want, you can draw manually also that diagram. That is not that much difficult. Along with this dendrogram, that diagram you
Hello,
can
sir.
draw manually also. Yes.
Yes, sir. I have one question.
Huh.
On what basis is genotypes divided in different clusters?
So see that last cluster dendrogram command line, the first line. Here we are
Yes.
here we are distributing the genotypes on the basis of this distance. Forget the rest. This come on this command, these genotypes are distributed. And this distance we have calculated here. This is that distance. So likewise, we will say like grouping of genotypes on the basis of cluster distance.
Excuse me, sir.
Yeah. Hello.
Sir, in this dendrogram, uh, how can we decide that from how much distance we will consider these clusters? We can also consider them as two here.
Cluster distance already you have calculated, na?
So the distance is given on that y 0, 100, 200, and 300.
So, but here already you have the values.
Okay, sir.
So, like on the basis of that values, you can interpret the dendrogram. So, likewise, now with
Excuse me, sir. Sir,
modified, we got 5. Yes.
sir, intra-cluster distance can be 0 also, what, sir?
Yeah, if single genotype is there, that maybe it will be come at 0.
And then if there is more than 2 genotypes, sir, then it can be 0 or not, sir? In any case, is it possible to have 0 intra-cluster distance, sir?
If more than 1 or more than 2 if more than 2 genotypes, means as far as I have not seen 0. If more than 2-3 genotypes are there,
Okay.
I have not seen. It may
Okay.
be, but I have not seen that case.
Okay, sir. Thank you, sir.
Hello, sir. So can you please explain the dendrogram again?
1 minute. I will repeat it again. 1 minute.
Okay, sir.
Yeah. So see, firstly, this is the command line for D square. So D square is calculation of genetic divergence within your germplasms. Yeah. And one more thing, you as a researcher, whenever if you are going for calculation of this genetic divergence across the germplasm, so that germplasms you need to collect from different regions. Like from each and every state, you need to collect some 1 or 2, 2 to 3 germplasms from each and every state. So it is, we are doing like we are collecting some germplasms from IRI or some research institutes. But if you will collect diversity, more diverse germplasm you will collect, so this diversity result will be good. Yeah. So for this diversity, we are using 2 files. One is name with cotton In that carton, we have replicated data, 12 genotypes, 3 replication data for 4 characters. And we have calculated the average for that, all the 4 characters. Okay. So, first is I will import the data. Meanwhile, those who understood clearly, they can calculate that genetic diversity from any replicated RBD data you have. So just calculate that average file, take the average file and replicated file and just go for the diversity analysis. Because you have a lot of replicated data, just you can try it. Okay, so this is replicated and average data I have imported. Then I have already run these libraries. Then after that, we need to, here actually we have this genotype replication factors and variables. So here we have to separate the variables. So for that, we have this command. So it is not 3:13. Here we have total 6 columns and our data is 3:6. And here we need to edit the file name, that is cotton. Then we need to go for multivariate analysis of variance. As we are doing multivariate analysis, D-square is one of the multivariate analysis. So we will set the model and simultaneously we will calculate the covariance. So here input file name is cotton. So here you will get the multivariate analysis of variance. After that, we'll go for percent contribution. What happened? Library, I have restarted the system. Library command. Yeah, so then this contribution. So likewise, percent contribution, if you have more number of characters, so then for each and every character it will show. Okay. So after that, here for Mahalanobis distance, we need average file. Name is already average, AVG. Here, to calculate the distance, here we have 2 methods. One is torture and second is modified torture with the addition of this algorithm. So simply copy this, paste. So here you will get 6 different clusters with the genotype number, which genotype is in which cluster, and cluster distance with intra and inter both. So which one is intra and which one is inter? From this table, which one is intra and which one is inter?
So diagonally it will be intra, means cluster 1 with cluster 1.
Okay, and
If one cluster 1 with cluster 2, it will be inter.
inter. So this will give both intra and intercluster. Then second is modified torture. So with this modified torture, instead of 6, here we got only 5 clusters. So then to draw the dendrogram, here we need to define the cluster. From this analysis, whatever clusters we have got, so based on this distance, this command line will distribute the genotypes. So this is the line. On the basis of distance, we need to define the clusters. As now here we have defined 6. So it will follow the normal torture method. If you will copy paste. So here you will get the dendrogram. So just cross check with that whether whatever the analysis you got, the number of means the genotype in cluster, so whether it is matching to this or not. If it is not matching, then check your command line. And simultaneously, for modified, here you need to define file. Yeah, so say 1, 2, 3, 4, 5, 6 with normal torture method. And with modified torture, these are the 5 clusters. So this is the cluster analysis.
Sir, for modified, why we need to put 5?
So these are the 2 methods. So whichever method you need to use, that is totally depend on you. Just like stability, we have 4, 5 models. So just like now methods are there.
Sir, I
Number
have a
of
question, sir.
clusters already we have calculated. Nothing to know in it, just we have calculated it. On that basis, we have drawn the diagram. So from this command, you got the 6 clusters. So from that, here you have the 6 clusters, 6 cluster dendrogram. Based on this inter and intra-cluster distance.
Excuse me, sir. Sir, what does that horizontal line indicate this in the STEM program?
Yeah, anyone can give the answer? So what is the maximum cluster distance value?
300 and something.
No. In our analysis. Here.
281, sir.
So
200.
actually, generally, red line you will see, it is the cutoff for the cluster height. So here you can see the cluster height. You are getting like this. So this is cutoff for cluster height.
Okay, sir.
Sir, excuse me, sir.
Ah, Dr. Sir.
Sir, I'm asking that if number of jumplogism is more and it is not in replicated data, so is there any possibilities to D-square clustering?
Non-replicated data.
Sir.
1 minute, we'll see that.
Sir, in that group K is equal to 9, we need to change that one. How many cluster we have formed, then we have to put that one.
Yeah.
For example, 6, then K is equal to.
Here you need to write how many cluster you got after analysis. Here and here at 2 places you need to write. How many are the clusters you got? Then, and as we are running this distance command very firstly, so it will calculate it will distribute the genotypes based on the cluster distance.
Hello, sir.
Yes.
Sir, instead of distance, we can use another different command to distribute these genotypes.
Generally, see, cluster analysis distribution will be purely on the cluster distance. So see, what you can conclude from this as a researcher or as a breeder point of view, what will be your final output or conclusion from this?
Like this genotypes is depends on or divided on the basis of this cluster like that.
Yeah, but what will be the why you are doing this?
For
516, 100% similarity.
Means this genotype which falls into one cluster means they have same origin.
Okay. And if in our case, this genotype 5 is falling in one cluster and genotype 12 is in 6th cluster.
highly diverse lines, sir.
So that's why we are distributing these genotypes on the basis of cluster distance. Yeah, and another thing that Dr. Saab raised with related to non-replicated data. So, sir, if you can see the script, that this is the standard format for Mahalanobis distance. So, if your data is non-replicated, it is not possible to go for this multivariate analysis and covariance matrix. So, if we will not analyze this MANOVA and covariance matrix, you will not get this cluster distance.
Okay, sir. But in other software like Darwin, the phenotypic data.
Yeah,
is
that
binary
diversity is different.
Yes, sir. Yes, sir. I know, sir. That's why I'm asking if any possibilities here, but you told that, yeah, really multivariate
That
analysis is not possible.
Darwin diversity is different. This is our completely our field data diversity.
Yes, yes.
This is purely standard format for Mahalanobis distance. That Darwin is completely different.
Yes, yes.
So can we perform this once for qualitative traits?
Yeah.
So can we perform this D-square analysis for qualitative traits?
Which type of traits you have?
Sir, like flower color and that qualitative traits.
So because see, as like for PCA we have tried, but actually these things we are calculating for quantitative traits only. If
Yes,
you
sir.
want to analyze, just you need to convert the data for your sake.
Sir, we like we did for PC, we coded them.
Because
Similarly,
in
we have to
standard,
code for D-square
as a
also.
standard procedure, it is not possible for qualitative data, but for your sake, you can calculate it.
So by converting them into codes as we did in PC.
Because see, as we are saying, this D square is one of the type of multivariate analysis. So
Yes,
we
sir.
are calculating covariance matrix. We are calculating the distance. So to calculate this
Okay, sir.
covariance matrix and cluster distance, we need numerical input to perform that covariance and MANOVA function. Compulsorily, we need
sir.
that numerical input.
Okay, sir.
So just you can try this now with different data sets. Means if in different data set, if you will encounter some error, that we can solve it now only. That I have shared some data sets like D1, D2, D3. Just you can take any data set and calculate.
Sorry, Prof. In that first model for DV, the very first one that you did, after assigning the columns, what else did you
I'm
do?
not getting your question.
I'm saying after the very first script that you ran after loading.
Here.
Yeah, the DV. After assigning 3 to
Okay.
6, what else did you do there?
Here you are saying what I did.
Yeah, after assigning the data set that it runs 3 to 6.
So
to 6
simple,
column.
simple is after running library, as here, I will show, as here we have replicated data, we have two factors like genotype replication, and after that our data is starting. Okay. So the first step we need to do is we need to separate our quantitative data. So to separate that, this command is there. So here we need to change our file name. That is in our case, it is cotton. And here I have written 3 is to 13. But when you will see our data, it is 3 is to 6. As total 6 columns are there, data is starting from 3 up to 6. So here you need to write 6 only. That's it.
Okay, thanks. That was the file name that I was missing.
Yeah, file name you need to change. Or if you don't want to change, then you need to annotate your file name with that data. Either way you can do that. No problem. So just take that D1, D2, D3, any file you like, and just perform this. And just check the dendrogram. How many clusters you are getting? But along with replicated data, you need to calculate the mean values. Don't forget to calculate the mean values and don't forget to import it.
Hello, sir.
Yes.
Sir, I have tried using the data set D1, but while running that percentage contribution code, I'm getting error. That is figure margins too large.
D1, D1, D1. 1 minute. D1. This D1, na?
Yes, sir.
So this data set you took, na? 1 minute.
Yes, sir.
So just give me 2 minutes. I will calculate the average for that. So, okay. Now I will show you with one data set. Yeah, so from starting, I will run the library. Just for errors, just see the screen. After completion of this, I will take the questions. I will import this D1. Okay. Then see the D1. So file name is D1. 3, 4, 5, 3, 4, 3, 4, 5, 6, 7, 8. 3:8. Okay. Then MANOVA and covariance. Here in place of genotype, as in our data set GEN, for replication REP, and data is D1. So this is covariance, right? This is the percent contribution. Yeah, now tell me which step you miss?
Sir, I'm not getting this pie chart.
So what I did so far, so which step you miss?
Sir, I have not missed anything. I have run all the code and library command as well. But I'm getting error. The error is figure presents too large.
Here you change the things.
Yes, sir. Yes, sir.
Here GENREP, here data file name.
Sir, I'm running it again.
Run it again. Raj.
Sir, we have to calculate average for this file as well. Sir, so how?
Yeah, just now
how,
I calculated.
Sir, sir, can you show it again, sir? How did you calculate average, sir? Sir, which from Excel, how we can calculate average directly, sir, like?
Not directly, just now I calculated in Excel. I will calculate for one more sheet. Wait. CL sir, just now I told like this. See this highlighted one. Like this, you need to edit. If you are using D1, D2, D3 data file, so here genotype is not there. Here it is written as capital GEN, capital REP. So you need to edit in that command.
Excuse me, sir.
Ah.
Sir, it is showing in my system, it is showing problem in installing BioTools. It is showing that
Then
this is not.
check BioTools again.
So it is saying
Just
that,
restart your RStudio and install it again from Tools option.
okay, sir.
Yeah, those who are not able to install the BioTools, restart the session and try with the Tools option. Yeah, so actually dendrogram it will display the genetic divergence, how diverse your genotypes are, as dendrogram will distribute the genotypes into different clusters based on the cluster distance.
Sir, it is saying that BioTools is not available for this version of R.
Okay, I will share one command line, just use that for BioTools.
Oh, okay, sir.
Just 1 minute.
Sir, that calculation of average also, sir, please.
1 minute. So just check this file, check this command line for installation. Before that, you need to run library for DevTools. DevTools, if it is not there, you need to install DevTools.
Okay, sir.
If DevTools is there, then run library DevTools, and after that, that command in I just pasted in chat box.
Okay, sir.
Yeah. So now see, after that, as I have calculated the average for D1, after contribution, I will start this Mahalanobis distance. And for D1, I got 8 cluster with normal torture. So Anjali, whether you have imported the average file?
For D1, sir?
No,
sir.
That's why that incompatible dimension is there. You need to calculate the average. Here, that's what I am showing screen. That's why that errors are coming.
Excuse me, sir. Sir,
Yes.
can we calculate this d square for L into T replicated data?
We have
Yeah, normal replicated data you can calculate.
Okay, sir.
But just the condition that your data should be quantitative, means numerical form.
Excuse me, sir. Sir, if the experiment is
Huh?
in augmented design where there is no replication, only mean data is there. So can we calculate the multivariate analysis?
So that's what I told earlier for calculation of multivariate analysis of variance. It is not possible to calculate only on mean. So just you can try this d square with this average file only. It will not come.
Okay, sir.
So with modified torture, I got only 3 clusters for D1. With normal torture 8 and modified torture 3 clusters. So both methods we are doing. So first I will draw with normal 8. So now see, this dendrogram is for D1 with normal torture method. And now I will draw for modified. Yeah, so see. This is for modified torture with 3 clusters. Yeah, so your errors resolve or they are still there?
So that average calculation, sir.
Average, so see, this is your file. Okay, so just you need to insert the column, not column, insert the row here. 。 So like this way I am doing, the other way you need to find out. So you can select the multiple rows by pressing the control tab. I don't know about Mac. I think function and control something will be there. Then at a time you can insert the row. Just you can calculate the average for one genotype. Just copy that and just select the first column of each mean and directly paste here. As here formula is there, so only we will copy paste the formula, so you will get this mean value. So after that, assign filter to replication, then 1 2 and this blank, so delete, so deselect it. You will get this, completely select it and delete it and remove the filter. I will do it. Wait. So see first. Don't do anything mistake. See first what I will do. Yeah. So now see, I have calculated the average. Now simply select this, copy, and in paste special paste value. Assign filter to replication. Deselect this. Completely delete this. And then delete the replication column and assign the numbers. Number or name, whatever it may be. Just you can assign. So hardly it will take 2 to 3 minutes. For medium data, for long data, it will take 5, 7, 8 minutes.
Sir, we are calculating cluster from average value.
Yeah. We are calculating cluster from average value, but we are using, along with average value, we are using this covariance. This covariance will be there from this multivariate analysis of variance. So by this way, your replicated and mean data, both they are interconnected. Haji, Dr. Sir.
Yes, sir. Sir, in some books or references, more of these discrete statistics, we have seen like circular diagram where cluster distance are
Yeah,
also
that's what I'm
given in.
saying, that octagon,
Yes, sir, yes, sir.
many other softwares like Windows Stat is there. So that Windows Stat is giving that octagon.
Okay.
So that
Okay, sir.
octagon is like, see, you can, even you can draw it. Just these are your 5 clusters. You can connect it. You can add the, so likewise you can connect it.
Sir, for calculating this one, so which we need to follow from replicated data, or we need to go for this both like that you are doing?
For what?
this cluster analysis?
Cluster analysis, we are using both data, replicated and mean.
So now earlier you were doing from only replicated data, no? So that method we need to follow, or both?
Both files we are using. That's what I told. Though you are saying like from this command, average file is there, we are using for Mahalanobis distance. But if you will see the command line carefully, you can see the covariance. Covariance we have calculated from replicated data, and it is part of we got this covariance from this multivariate analysis of variance. So whatever the cluster distances we have calculated, that is both combination of replicated and average value. Yeah, so other number errors resolve.
Yes, sir.
So this is dendrogram. Then open that violin plot file. So here also mean data is there, like two different populations and number of genotypes, and these 3-4 characters are there, and this is the violin script. Just it will start with the, just first import that data. Okay, then initiation. I have selected one character, that is PPPB. You can select any other one, no problem. Then in 2 step, you will get the violin plot. It is integration of box plot and violin plot. So here on x-axis, the types of population, and on y-axis, the observation. So this type of graph is, it will be easy to visualize if you are running some crossing blocks or crossing progenies. If you are evaluating, So in that case, these representations will be quite helpful. So if see, we will do it on different data set. Just you can try this. I will use some different data. Yeah, so likewise, different data set I will share with you. So this is the violin plot. So it is used to compare the progenies of different types of crossing populations. Like here you can see BC1F1 from 17 to 24, 25, 26. So different populations are there and different number of genotypes are there. So from that basis, this is the visualization. Okay. So everyone is having ICI mapping in their system?
Yes, sir.
Yes, sir.
Just give me a minute, I will connect from different system. Now for this QTL mapping, I need to work on two systems. I will share one data file in WhatsApp group. Yeah, so from group, just download that Excel file. Arabidopsis rel. And meantime, just to see. Again, I will share. Yeah. So just open that LT data. LT data set. So first tell me what is line and what is tester. Yes.
Line female and tester male pair.
Okay. So what is the benefit of what are the methods of estimation of combining ability? What are the different methods?
Sir, line into tester, diallele, partial diallele.
Okay. So generally, which method we will use?
Line into tester.
Why?
So by this we can know about the GC and ACA about specific.
Yeah, just elaborate your answer. It is correct, but just elaborate it. What specific?
So like for maximum heterosis, we require both GC and SCA. So we can calculate both of them together with line into tester method.
Here tester are few in number, sir.
Yeah, but so see, if you can see the data, you can see like each line we are crossing with each tester. So see line 1 with tester 678. Line 2 with tester 678. Likewise 3, 4, 5. Okay. And then what is this? These are the lines, data of line and data of tester separately. So likewise the arrangement. So I will show you for one character, you can estimate it for full data.
But sir, for line new tester analysis, 10 genotype minimum should be there.
Yeah, genotypes are, see, 5 total. I am giving just example.
Okay,
This is just a demo example data. You can calculate for your whole data. Same script will be same. This is this type of data arrangement you need to make. And code is very simple, 3-line code. As you already, I think, have the package Agricoli already installed in your system, just you can check with by running the library code whether it is there or not. If it is not there, just you need to install it. And import the data file. So this Agricoli is there in everyone's system. Okay. Yeah, so as I think during stability, AMI stability, we have installed it. Yeah. So then directly you can run this output command. So just here you need to edit what is the file name that you need to write here. What we need to analyze is line into tester. First is replication, second is line, third column tester, fourth column is yield. Yeah, and one more thing, if you will type this Agricoli R package in Google, you will get the full PDF. What are the different types of analysis you can do through this Agricoli? So all the analysis with short command lines, everything you will get in Google. If you will type Agricoli R package, PDF is there. I will that also I will show you. So like this, see. So this PDF is there. So just you can try this. All these different types of analysis are there with command lines. See, NCD 123, if anyone is working, we'll take it, but by your own you can try. Just you can download this. Likewise, Metan also. Where we use Metan package? We use Metan package for which purpose?
Stability, sir.
GG by plot.
Yeah, for that Metan itself, it is multi-environment trait analysis. So for Metan also, you will get the PDF. In that also, you will get the different types of analysis you can do from multi-environmental data with this kind of script. And with different with different stability indices also you can calculate. So all this, everything you can try by your own. Everything is available. Nothing is like rocket science. The more you will practice, more you will learn. That is the only funda. See, likewise, fast way to create the bar plots. Heading is fast way to clear, and 1 page to 1.5 page command lines are there. So likewise, just you can download this and you can use it. Don't just download it and keep in your system. Don't increase that space. Yeah, so after pasting this editing, just enter it. You will get all the combining ability parameters. With ANOVA, line into tester, analysis of variance including parents. Then this GCA effect, SCA effect, Then these are the standard errors for line tester, SCA, everything. Then these are the genetic components for line tester and average. And at last you will get the contributions. Contributions of lines tester and L into T. So with this single command line, you can estimate these many things. So you can edit this command if you have more number of threads. So you can edit it, no problem. So likewise, this is the short command of combining ability analysis by using Agricoli package. Okay. Then, so I think here any difficulty or error? In detail we will see this again, so don't worry about it.
Hello, sir. So can you please one time tell the formula or the code in which we had imported the data from text file to Excel? Or is there any code that you have shared? I don't
Code
remember.
is there. Code, I think it is there with you only with the file name is, I think, HapMap to Excel.
Okay, sir. Okay.
If I am not wrong, that file name is HapMap to Excel. If it is there, you can, I can share you 1 minute. I think it is there in group. Yeah, it is there in group only.
Okay, sir. I'll try once and will revert.
Just you can download it.
Sure, sir.
So one more thing. So lastly, I will show you linkage map, how to draw the linkage map.
Sir, I am getting error in that Boolean script.
Darwin, D square.
Boolean script.
That you have shared, na? So you have to
Yes, sir.
draw as per the crosses, not genotypes.
Sir, I am getting error also by running as per class.
Your number? This 7837?
Yes, sir.
Team set. Have you run that ggplot too?
Yes, sir.
Have you imported the data?
Yes, sir.
Okay, Ylene. Yes. Stop. Do one thing, restart that session and do it again.
Okay.
Yeah. So remaining people download that Arabidopsis file. Okay. So I am presenting the screen with name Ajay. Just see this. Yeah. So this Excel file is visible.
Yes, sir. Yes, sir.
Yeah. So see here you can see in one Excel file 3 sheets are there. In first file, general information. Second file, numerical genomic data, genotypic data that we have seen how to convert it into numerical form, either through tassel or through R command line. So this is that data. This is the list of markers. Okay. And then at last one sheet is there, anchor. Markers are there, but after analysis, in output file, we will get the anchor numbers. Okay. So, and in this general information, you can see as this is the data for real population. So this mapping population type is 4. For different population, different numbers are there. So as now we are handling real, so mapping population type for real is 4. Mapping function, as there are 3 mapping function. One is Kosambi mapping function, second is Halden mapping function, and third is Morgan mapping function. So this all we will see one by one in detail. Just today I am showing you how we'll draw the diagram. Then marker space. Marker space means how these markers are displaying in your genotypic file. So here we are giving the positions of the markers, not interval. So position means serial number is 2. Number of markers are 64. Number of markers are 64. And the size is 120. So number of markers are 64. Size is 120. So this is the input file format for linkage map construction in ICI mapping. So after that, I will open this IQTL ICI mapping software. So anyone is getting error in installing this?
No, sir.
Sir, one question,
Everyone
sir. That file is we generate through Tassel, sir.
You can generate through tassel as well as through R. Already we have seen how to convert it into numerical format. Already we have seen, I have shared codes also. So tomorrow again we will see that, don't worry. So this is the ICI mapping software. So very first step, you need to go in file, create the project. Okay, so here you can enter any name. I will enter today's date. Just okay. So second step is we need to construct the linkage map. So for that, we need to go to our folder. And here we are dealing with XLSX file. So we will import it. Okay, already it is open. I need to close that. So this file I have imported, so it will look like this. This is the input file. We have imported the Excel. After importing, it will convert it into map file. Okay. So here in at bottom you can see some options like parameters, threshold value. Then this first step is grouping. Second step is ordering. And third step is rippling. So likewise, steps
The
are
map
there.
file.
First, yes. So steps are there. First is threshold value. Here we need to set by LOD. So what is LOD?
Logarithm of odds.
Logarithm of
Odds. So probability of marker linked together upon probability of marker that are not linked together.
yeah. So here you need to keep the value as 5. Okay. Insert and then you need to call for grouping. In second step, you need to select the option called serration. So why, what, where? So I will tell you tomorrow in detail. Serration. Again, LOD. Then you need to order. And here window size 5, LOD, rippling. And then here just you can click everything. We'll just see the mapping files only. But you can click everything. So it is, you can see here. So after completion, if you will click on this plus sign results, you will see this lot of files. So then just you can double click it on this map file. And if you will scroll down, so you can see the data. And just click on this, double click on this result. So here you will get the option. These are the chromosomes. So likewise, this is the linkage. This is called as linkage map. So how to create that files? What are these all steps that I will tell? Don't worry. So on left hand side, the positions are there. Right hand side, marker names. You can change it here. See marker name. You can assign to left. Positions you can assign to right side. Okay. So while running here, if you have selected this everything, first I will clear this results. Yeah, so this is the input. Grouping, ordering, rippling, output. Okay, so these files will come. So now, yeah, so here you will get generally the positions of the QTLs associated with it, or positions of markers. So that same this linkage map you need to use for QTL mapping. So as I will tell you one demo file, I will show you. So for if it is real, then we need to go for QTL mapping in biparental population. If you have data of multi-environmental data, if you have, then you need to go for MET. NAM population, NAM. So all these different type of files will be there. So as of now, we will go for this biparental. So here examples are there. If you have installed it yesterday, day before yesterday, I told you, example files will be there. So randomly, I will select any example file and I will import it. So see, here you can, if you will scroll down, you can see this number of chromosomes, 10 chromosomes, this number of markers across the chromosomes. These are the marker list, these are the chromosome numbers, and these are the linkage map distance, what we have seen in our linkage map. This distance we need to include in this biparental population file. And then at last, the numerical data, and below it, we need to incorporate phenotypic data file. So as far as the parameters are concerned, parameters no need to change, as missing phenotype during analysis automatically they will delete. Then these thresholds already you need to keep as a default. This mapping method. So here total 6 methods are there. What is SMA?
Single marker analysis.
Simple interval mapping.
Then ICIM? ICIM, EPI?
Inclusive composite interval mapping.
Yeah, so I will select 2-3 methods. Already exist. Okay, no problem. So 3 methods. You can select more if you want. Then I will start this task. So we will see. So this type of file arrangement we need to prepare. So as we have genotypic file, we have phenotypic file, we can convert it into numerical data. So we have to arrange this file in this format. So still the analysis is going on by all the 2-3 methods. So as we have given the methods, like we have given epistasis also, we have given single marker analysis, we have given interval mapping also. So that's why it is taking some time. So before starting in detail, I will give one brief theoretical introduction of QTL mapping and methods of QTL mapping, so that will be easy. So in short, basic things I will tell, then immediately after that I will start this. Yeah, so see, now I think it is completed. Yes, run complete. Yeah, so now if you can see this here. Yeah, so as maize is having 10 chromosomes, so you can see the chromosomes markers positions. Here you can see the LOD. And here, I don't know whether you can see the significant QTLs. Red, highlighted in red color. Red color mark is there on that map.
Yes, sir.
Here.
Yes, sir.
Yeah, and then as we have given the additive ICI and MAD, so just see. With additive effect and LOD score. Next chromosome. And this is for all chromosomes. Position in whole genomic region, or you can say whole genome. For all traits, if you want chromosome names.
Sir, what about R square value, sir?
You just if you will check the output files in that everything is there. That we will see, sir, one by one each file we will see. No problem. In tables you will get, sir, no issue. So this is additive. Then for epistasis, it will give like this. Yeah, so this is for one trait. So as this is demo data set, you will not find any interactions. As tomorrow we will arrange it, so interactions will be there across the chromosomes. And with this linkage map, here easily you can save it. For saving, just you need to right click. Either you can save as image, or if you want in JPG, from here you can change the format. You can save it. Where it is. Yeah. So like this, it will be visible with LOD score. And with significant QTLs. Wherever significant QTLs will be there, this type of highlighting will be there. So these things we will start from tomorrow. Is QTL mapping. First we will complete this ICIM. Then in R also we can do. R I will show you. Yeah, so this R is visible.
Yes, sir.
Yeah. So for that purpose, you need to install one package. That is called as qtl.gci mapping. So this QTL mapping is actually, it is not based on coding. So I will show you the GUI once. What happened? I think something is there. Just download this package. It will work. No problem. That name I have pasted in chat box. qtl.gci mapping. Wait. I got the error. It is not that. It is with GUI. That's why it is not coming. There are two packages. Yeah. So like this GUI is there. So go for this second, not this first. So this type of GUI is there. If you will start, so here you can do with GCIM, WinQTL cartographer is also there, and QTL ICI mapping is also there. So all three are there. I will show you with one demo data set. Yeah, so see, like this format will be there. This is the markers and these are the genotypes. And then we need to go to parameter setting as it belongs to double haploid. So this double haploid population. Then here we need to set the path. Okay, rest is same. Just press run. And wait for the yeah, so it is like this. We will see that this tomorrow. ICI mapping and this QTL dot. Yeah, so this running is completed. So after that, if you will see it here, you will get this type of graph with the result. Only significant QTLs you will get. Yeah, Dr. Saab, here you will get your R square value. Then with LOD, left marker, right marker, everything detail, we'll see tomorrow. So please join on time. Don't be late. Ah. Yes, madam.
Excuse me, sir. Sir, I have two queries related with the assignment. So first one is I'm not able to do data cleaning by R code. So for
What? Come again?
So for GWAS analysis,
Huh?
I'm not able to do data cloning by R coding. I'm just using the cleaning by the TESS software for the genotyping.
No problem. That we will repeat it again. No issue.
Okay,
Whenever
sir.
we will get time, we will repeat it.
Okay, sir. And the other thing is like when we map, when we map the QTL, like JAVAS for when we done, then we have to do with the phytosome. We have to we work with phytosome, and then we have to find the is it like available on software, available on the
Yeah.
data set or not. So what's the crop is in this file like?
So this both whatever both genotypic file I have shared both belongs to sorghum.
Okay, sir.
Yeah.
Thank you, sir.
Okay. So tomorrow don't be late. Join on 6:15. Tomorrow we'll start at 6:15. Okay. So thank you guys. Tomorrow we'll start this in detail QTL mapping. Okay. Thank you.
Thank you, sir.